Alright, so in this video, we are going to look at biology paper 2. So the instruction is saying, answer all the questions in this section in the spaces provided. So we have the first question. Question A. The diagram below represents a plant in the division Biophyta. So we have part A, B up to D. So now, Roman numeral number 1 is saying, name the parts labeled B and D, 2 marks. Now, Biophyta, these are qualified to be in the family of the Rhizopus. They share some common features, I may say. So, when you talk of structure A is not a shoot, but rather a capsule, or we can just say a spore case. Then when it comes to structure B is called sporangium, the same structure that the Rhizopus has. So B is called the sporangium. Then when it comes to C we can say the main axis. Okay, so C is called the main axis or the leaf-like structure. So it's the main axis or the leaf-like structure. Then when it comes to D, of course D is not the root but rather the rhizoid. So B, I'm saying that it is the sporangium. D is called the Rhizopus. Then for Roman numeral number 2, it is saying state one function for each of the parts labeled A and C. So as you can see here, I'm from saying that A is the spore case. Okay. Then as for C, it is the main axis or the leaf-like structure. Now they are asking us the function. So what is the function of A? So when it comes to A, for A, we are saying it is the spore case. What is the function of the spore case? The function of the spore case is to release the spores during fertilization. Then when it comes to C is this part A, the leaf-like structure. So the function of this one here, it is to carry out the process of photosynthesis to produce food for the plant. Okay, so those are the functions. The next question, B. The diagram below represents a member of the kingdom Animalia. Okay, we have Roman numeral number 1. Name the phylum to which the organism belongs. So there are 3 things here that you need to understand. There is what we call the kingdom. There is also what we call the phylum. There is also what we call the class. So as you can see here, this one is the grasshopper. So the grasshopper is found in the kingdom of Animalia. Okay. Then when it comes to the phylum in which the grasshopper is found is called the arthropods. Okay. It is found in the phylum called the arthropods. Then when it comes to the class, the grasshopper is found in the class of Insecta. It's more like an insect. Why are we saying an insecta or an insect? It has 3 legs or 3 pair of legs, I may say. So we have 1, 2, and 3. So it has 3 pair of legs. The next question. Using observable features in the diagram, give 3 reasons for the answer in B Roman numeral number 1, and it is scaling 3 marks. So, as you can see here, why are we saying this grasshopper belongs to the phylum of arthropods? So those are the reasons which they need in this question. So the first one, we are going to say they have segmented body. When you talk of segmented body, we are talking about separate bodies. The first one, they have the separate head. It is segmented. Apart from that one, they have a thoracic here, Apart from that one now, they have also the abdomen. So those are the segmented body parts of the grasshopper or this arthropod. Then another reason we are going to say, since this one is an arthropod, it is going to have what? An exoskeleton made of chitin. So this part here that you can see here, it's made of the exoskeleton. Now, the same exoskeleton, it is made of a material called the chitin. Chitin, it is a polysaccharide. Okay? Then another reason we are going to say, this one, it is an arthropod, which have what? The jointed legs and the antenna. So in top of the antenna, there are these. These are called the antennas. And as you can see here, the legs, they are jointed. So those are the reasons why this one classified to be an arthropod and belong to the class called the Insecta. So let's see the next question. The next question is saying figure 2.1 shows some of the processes and organs involved in the regulation of blood. So here we have the dietary intake, the blood sugar, apart from the blood sugar, process A, process B, and so on. Now the first question is saying, name organ C, which detects changes in blood levels. Okay, let me just pause a bit here. According to this question here, they are asking us to give them the organ which is able to detect the changes in the blood sugar. When you talk of blood sugar, you talk of the level of glucose in the blood. Or I might say, they're talking about glucose. Is the glucose high or low? Now, the organ which is going to detect if the glucose is high or the glucose is low, which organ is that one? So as you can see here, we have organ C. The work of this organ C, we have these arrows, this one and this one here. For this one, we have this one which is called the insulin. Then for this arrow here, we have this one called the glucagon. So now, which organ is capable of releasing glucagon and insulin? Of course, it is pancreas. So the answer here is just going to be pancreas. Let's see the next question. The next question is saying, what effects do glucagon and insulin have on the amount of glucose in the blood? The first one, glucagon. Now, mind you, this question is asking about the effect of glucagon and insulin. When it comes to glucagon, you're going to find that for glucagon to be activated, it is only activated when there is less glucose in the blood. So the effect of this glucagon, it is to increase the amount of what? Sugar or glucose in the blood. Period. Now you're going to find that when you're having a lot of blood sugar or high level of blood sugar in your body or in blood, what is the effect of insulin? Insulin is going to lower. So let me just say, decreases the amount of sugar or glucose in the blood. Then as for question C, processes A and B occur in the kidney. Name. Number 1, process A, in which sugar leaves the blood. So let's come back to the diagram. So we have this A here. The sugar is leaving the blood. So what process is that one? The process is called the ultrafiltration, and this ultrafiltration occurs at the point called glomerulus. Once it is filtered out, it is going to be called as the glomerulus filtrate. Then for process B, number 2, it is saying process B, in which sugar re-enters the blood. So let's see on the diagram here. So sugar is re-entering the blood. What process is that one? The process is called selective reabsorption, and this selective reabsorption takes place in in the part of the nephron called the proximal convoluted tubule, or we can say tube. Then for question D, it is saying diabetics lack insulin and are treated by having regular injections of insulin. Explain why this hormone can't be taken as a pill. So in talk of hormones, they are just usually proteins. Okay, so since insulin is a hormone and it is in form of a protein, if taken as a pill, you're going to find that it is going to be digested by what? The protein enzymes. So the reason why this one can't be taken as a pill is that it is going to be broken down by the digestive what? Enzymes in the stomach and in the intestines. So that is the reason. So once it is broken down in the stomach or in the intestines, this means that it won't reach the bloodstream in the active form. So it must be injected to be effective. I guess you have gotten my point there. So the question is saying the diagrams in figure 3.1 show demonstrations of two simple processes. So we have A at start, we also have B at start, then after 1 hour, even here after 1 hour. So as you can see here, the changes, we have a colored crystal inside here, then after 1 hour, it has spread at the bottom here. Here we are having fewer liquids in the Viking tube or dialysis tubing. Then after 1 hour, the dialysis tubing has increased in size. The first question, name the processes demonstrated in figure 3.1, that is A and B. So when it comes to A, this one here is called the colored crystal. When you hear the word colored crystal, or things deals with colored substances, that one deals with diffusion. Okay, so meaning for process A, we are going to say this one is going to fall under diffusion. Then as you can see here, we have a dialysis tubing. A dialysis tubing is acting as a selectively semi-permeable. Whenever a selective semi-permeable membrane is involved, that one is going to fall under osmosis. So we have A called diffusion, then B called osmosis. Question B, Roman numeral number 1. State two changes visible in B after 1 hour. So we need to state two changes visible in B after 1 hour. So as you can see here, the only changes which I can see in this experiment after 1 hour, as you can see here, the dialysis tubing has increased, or we can say the liquid level inside the dialysis tubing has increased. As you can see here, the level of liquid at start was fewer compared after 1 hour. After that, as you can see here, the level of the liquid or the volume of water in the beaker at start was high compared to the level of water after 1 hour. As you can see here, it has decreased. So we are going to say the volume of water in the beaker decreased slightly. For Roman numeral number 2, it is saying, explain how the changes in A and B are brought about. So let me start with A. A, we have said that it is diffusion, and then B, we have said it's what? Osmosis. Now, as you can see here, colored crystal is highly concentrated. Okay? Colored crystal is what? Highly concentrated. So you're going to find that this colored crystal can be anything like a dye molecule. When I say dye molecule, I mean about this spelling for dye molecule, not diatomic, but the dye molecule. Okay? Now, we can start by saying the dye molecules in the crystal at a high concentration, which means this one here, it has a high concentration, okay, at the start. So as you can see here, at the start, it is going to have higher concentration. So since it is having higher concentration, meaning you're going to find that it is going to spread or move randomly into the surrounding water, spreading out until evenly distributed, okay? Now you're going to find that, as you can see on this diagram here, no selective semi-permeable membrane is involved. So you can just say no membrane is needed. So I think I've made myself clear on that one. Then when it comes to B, there are 4 terms, or I may say 2 terms, which are used under osmosis. There is what we call higher outer potential and lower outer potential. When you talk of lower outer potential is a concentration with more solutes. Then when you talk of higher outer potential is a solution with less solutes in it. So as you can see here, by definition, in talk of osmosis, the movement of water molecules from the region of lower concentration to the region of what? Higher concentration. So inside the dialysis tubing here, we have this solution here, sugar solution. So meaning inside the dialysis tubing, we have higher concentration, or I may say lower water potential. So in this case here, we're going to say the sugar solution inside the tubing at lower water potential. When I say lower water potential, that is higher concentration. So than the pure water outside. So we're talking about this pure water outside. So water molecules moved across the partially permeable membrane into the tubing to balance the concentration. Now, after balancing the concentration, you're going to find that this is going to cause the liquid level inside the dialysis tubing to rise. So this is what has happened here. For us to reach this stage here for osmosis, and for us to reach this stage here for diffusion. So that was the last one. Thanks for watching.